Um, Thank you so much for the invitation. I'm super excited to share some of my work I have done as a trainee in UGA supported by PGCOE. So everyone can see my slide, right?
Yes.
Cool. Today's my talk. The topic is tracking local scale viral transmission using genomic data. Um, it's a key word about today's talk is pathogen genomics. So why are they so important, and why do we study viral genome for epidemiological inference? So SARS-CoV-2, for example, like other coronaviruses, has a built-in error correcting mechanism. Because of this, it mutates more slowly than virus like HIV and influenza. But even with that proofreading, SARS-CoV-2 still evolves relatively high rate. That's more than two substitution per month. And these mutations accumulate over time in ways that are both measurable and trackable. So by studying this viral genome, we can monitor the emergence of key mutations and perhaps most powerfully use the evolution history to uncover unobserved transmission events. Another key word is local scale. Why do we focus on local scale? Well, the global spread of SARS-CoV-2 triggered new outbreaks across continent and countries. The majority of the case actually rose from local scale. Um, at the same time, academic and public health lab around the world, they have a Herculean effort to gather this viral genome. So it's at a scale that far surpasses any previous outbreak response. So there are over 17 million whole genome sequence uploaded and shared in Gazette. So we're facing a major challenge in here, but there's also a remarkable opportunity that is uh, how to utilize this sequence to understand more quantitatively about transmission. Um, so this is understanding local transmission dynamic in a quantitatively way is essential for designing effective prevention strategies. For example, like identify risk groups and implement target control measures. So this kind of work directly benefit local public health efforts, whether at a state level like Georgia or city level like Athens, even like a community level in UGA. Um, Bayesian statistic method has long been considered as a golden standard for inferring evolutionary parameter from genomic data. So as a core of the Bayesian analysis, they have two key components, is the data and the model. So the model describes the process that generates the data. For example, we have the demographic model that describe population level process. The side model, which count for the variation in substitution rate, and the clock model translate genetic difference distance into time scale, allowing us to estimate when the event occurred. So use a base theorem, we calculate the posterior distribution, which is a proportion to the likelihood of data given the model multiplied by priors beliefs. So one of the key strengths of Bayesian master is that flexibility. They allow us to incorporate like multiple models and source of uncertainties into a coherent framework. This approach has been successfully applied to study global circulation of viruses such as Zika, Ebola, and influenza. However, the sheer volume of SARS-CoV-2 genome presents significant computational challenge. So when working with dataset, for example, that exceed 5,000 sequence, both the burn-in period and the computational time acquired is really, really long. So joint estimating this parameter is computational infeasible. requiring them to be estimated separately. Um, so in the past 5 years, we see a surge in development of this model and bioinformatic tools designed to extract the valuable insights like to dig gold. Uh, so uh, there have been tremendous effort in developing these new tools and it's continue. Um, so this is a booming time for master development, but there's still a clear need for further improvement in computational approaches. Um, Reducing the time lag between sequencing and inference is critical. So fast real-time analysis makes genomic data far more actionable and meaningful. The following of the talk today is some of my PhD works and some of the new the pipeline developed to help trace the transmission. So first part is a case study in Texas, and the title is Characterizing Spatial Epidemiology in the Heterogeneous Transmission Landscape using the Spatial Transmission Count Statistic. We focus on the state of Texas, and the population structure and distribution in Texas is not homogeneous, with most of the population centered in a very few large and well-connected urban centers, surrounded by many smaller rural communities. On the map of Texas, counties are color-coded based on their corresponding RUCC. So RUCC1 representing the metro regions with population more than 1 million. So these are the most populous metro area in Texas, which are Dallas, Houston, San Antonio, and Austin. Excuse me. So rural population in Texas has been significantly affected by COVID-19. Unfortunately, civilian effort have predominantly focused on urban center, leading to limited investigation into the rural area in the circulation of SARS-CoV-2. Um, Previous phylodynamic analysis has shown the frequent bidirectional transmission occur between rural and urban. However, very few studies investigate the differences in transmission pattern between these areas. So the core question uh, in this study is how regional heterogeneity, such as rural versus urban, shaped viral spread and epidemiological trajectories. So the key challenge is to quantify transmission patterns so we can easily compare them. So let's take taxes as an example. So transmission event related to taxes can be classified into three categories: imports, local transmission, and exports. So given the phylogeny, we begin by assigning geographic information to the nodes using the ancestral tree reconstruction. Then, by comparing each node state with its parent state, we classify branches into such categories. So in this example tree, we identify 6 imports, 9 local transmission, and 3 exports. So guided by the viral phylogeny, we summarize weekly count of local spread, viral inflow, and viral outflow. To quantitatively compare transmission trend between regions, we introduce these two metrics. One is local importance score, measures the proportion of new case resulting from external introduction. This value range from 0 to 1, with higher value indicate greater reliance on external introduction. We also have the source sync score, represents the net viral flow, normalized by epidemic size. So the value range from -1 to 1, with positive value indicating a source-like behavior, and the negative value indicating like a sink-like behavior. Um, so in genomic epi, analyzing subsampling biases are very crucial steps. So we calculate the sampling ratio by dividing the number of available genomes by the number of reported case to explore sampling uh, biases. So in this figure, each dot representing the sampling ratio in the specific region, and we observe significant heterogeneity in sampling ratio across subregions from epi-week 14 to epi-week uh, uh, 43. To mitigate this sampling biases, we applied a subsampling scheme. So we adopted a sampling ratio of 0.006 as a baseline. All the region undersample, all the available genome were retained. Otherwise, more oversample region or downsample to match the baseline rate. So as a result, we select over 5,000 text genome, and you can see the variance in sampling drops substantially. So in total, we have a big genome database with over 5,000 taxa sample and over 6,000 global sequence. So one of the first questions is we want to know what does the epidemic in Texas look like. We're presenting here is a timescale phylogeny with branch colored as imports, local transmission, and export. So this compact tree has more than 10,000 tips. Very hard to grasp information from it. And using our, oh, sorry, using our transmission count statistic, we show the epidemic trends in Texas in a clear and simple way in here. So the initial introduction began at late March, the epidemic peak occurring around July, and Texas has multiple imports and export to the global epidemics in here. Okay. The following question is we want to know what caused this mass scale of local transmission in Texas. Where are the source of viral spread in Texas? We show the phylogenetic network of Texas. Uh, uh, so all the nodes in here are the metro areas. In this network, each node represent a metro area, and the width of the edge is proportional to the spatial transmission count. So centrality analysis ranked this metropolitan area as based on their betweenness, closeness, and degree centrality. So these four major urban centers, Dallas, Houston, San Antonio, and Austin, um, are suggest populated urban center play a critical role in the viral spread across Texas. On the panel B is uh, uh, this conclusion is further supported by panel B, which is using our sourcing score. So we summarize the epidemic trends across 25 metro areas and their surrounding rural region, and then calculate the cumulative sourcing score for each. So this shows highly populated urban centers are the major source of Delta epidemic in Texas. Then we quantitatively compared the epidemic in urban center versus rural area. So using the Houston as a representative city, we compare the epidemic trends in urban center to those in the rural area. Let's break it down. So first, we found Houston as a highly connected and large urban center served as an important hub linking the outbreak across Texas to the broader global pandemic. Then, using our local importance score, we found out the epidemic in Houston in the urban is more locally maintained because they have a smaller local importance score. Lastly, we found out Houston function as a viral source, rural as a sink based on the source sink score, and the role of the population as a source of sink really evolved dynamically as the outbreak progress and the host immunity develops. So, uh, for as a summary, we find out that urban centers serve as a key hub linking the local outbreak to the global pandemic. So introduction from global source for extensive transmission within these urban centers. This urban outbreak subsequently seeded the spread of virus to the other part of the state. So rural areas primarily act as receivers, and outbreak were largely driven by external introductions. So this is a takeaway point of this study. We developed a pipeline to understand local scale transmission trend. So the approach include proportional genomic sampling based on the case count, followed by the phylogeographic analysis using a workflow like next string workflow. Lastly, we summarize and compare the transmission pattern across the subregion using our metric, the sourcing score and local importance score. Now, let's go to the second part of the study. Another study, we focus, instead of the whole Texas, we focus on city of Houston. So Houston is the largest city in the southern United States. It's also most economically segregated city with lots of disparity in income, education, and occupations. The research goal in here is to reconstruct disease spread by identifying viral introduction and tracing the dispersal of transmission clusters. We do have a new pipeline or algorithm to detect, to extract a locally circulating cluster. So imagine you preparing your data set and you place focal area sequence in the context of a proximal background. Then we can construct ancestral location of the virus as Houston and non-Houston. Then we identify introduction events on branches connecting the non-Houston nodes to the Houston node. The introduction time was defined at the midpoint of these branches. Starting from these introduction events, we apply a depth-first search to identify and encompass all the downstream node or tips assigned to Houston, grouping them as a circulating cluster. Then we can refine this cluster by pruning some of the branches, and finally we obtain this cluster. So this is a workflow we developed, and we will apply this to the following study. Okay, so let's talk about the data set we have. So we build an even bigger data set with over 30,000 genomes, including 10,000 from Houston. Anyway, we identify over 1,000 of introduction events, and all these introduction events are highlighted as yellow dots. So each introduction events result as a locally circulating clusters. To illustrate this, so we highlight some of the point, and these are some of the just show as an example. There are the resulting locally circulating clusters. The tips are colored by the counties in Houston. This is just an example. Okay. What we found in here is earlier introduction were more likely to result in larger clusters. So we classify this cluster, 1,000 clusters, by their size, and we found out the green line, which representing the cluster size more than 10, showed up earlier. We compare the introduction from domestic or international, and we found out most of them are coming from domestically. This showing Houston is unlikely like a hub, the first hub for SARS-CoV-2 introduction to the US. So this is so we select some of the large cluster, like 82 cluster. Each of them contained more than 10 isolates. We further analysis the local spread. The local spread meaning here is the spread within Houston. So all these clusters include more than 6,000 sequences. So we investigate the correlation between phylogeny structure and the demographic traits, yeah, the age and sex. In here, I want to show, so this result really show viral transmission is more constrained within the age group than within the sex group. We further quantify the transmission rate between age group, and the infected adults were significant source for other age groups. So the final piece of the analysis is really to understand the viral dispersal in subregion and their heterogeneous dynamic. So So we characterizing local traces into different 9 counties. The joint model identify 14 transmission supported by the base factor. In here, we we really show the heterogeneous dynamic. So the Harris County stand out as a viral source in here. They have the very low local importance score and very long persistent time. So targeted public health intervention in this identified source region uh, could have a broader impact to reduce the spread of virus across greater Houston area. Okay, the takeaway point in here is really we're presenting this and this workflow to trace imported cluster through community. So our approach point when, where, and how many introduction occurs while tracking the circulation of resulting clusters by incorporating metrics as sourcing score, local importance score, and persistent time. Our analysis revealed transmission heterogeneity between sub-region of focal area. So this is I'm 1 minute going. I need to stop myself right now, but I want to thank my former lab. I want to thank Dr. Ball, the support from him, and everyone who support me carrying this research. And I will stop right now and open to any questions.
Feel free to use the chat if you have any questions as well. Okay. Looks like we may not have any questions, at least not right now. So how about we switch over to Dr. Anderson's talk or sorry, Dr. Morrison's talk, and then we can come back at the end for questions for both. All right. Thank you so much, Leke. Dr. Morrison? You're on mute, by the way.
All right, can you see that?
Yes, we can.
All right, thank you guys for having me here today. I feel like these are very complimentary talks, so I'm excited. Okay. All right. And so just to kind of be clear, I'm an epidemiologist, so I am not a laboratorian. I'm not a bioinformatician. You won't hear me talking at all really about pipelines. I do know that some of the dengue sequencing we do uses a pipeline called Daytona, but that's about as much as I know on that piece. But I'm actually here to provide kind of a slightly different perspective on how we use genomic data as part of some of our disease investigations. But to kind of look at this from the big picture view, why do we even do surveillance? And it's really to try to prevent large disease outbreaks, and we do that by targeting our control activity. And so you need to know, where activity is occurring, when is it occurring, who is it impacting, so that you can target that activity appropriately. And for, primarily going to be talking about mosquito-borne diseases today. For that, there's different ways of control that we are considering for this. There's things like vector management, so mosquito control, managing those mosquito populations that are flying around, whether it's using larvicide to kill the mosquito larvae or adulticide to kill the adults that are flying around. Control could be thought of as being, increasing awareness in the medical community so that they think of these diseases, and the public as well, so that people can take the appropriate kind of personal protection measures to avoid even being bitten by mosquitoes in the first place. Draining standing water, wearing long sleeves and pants, and mosquito repellent and things like that. So control is really one of the biggest things that we do, because we have these direct public health actions that can be attributed to the surveillance activities that we are conducting. And so again, kind of looking at it big picture, you know, there's a lot of barriers for us to even find out about a person. They have to be sick. So not all mosquito-borne diseases is someone actually symptomatic. They have to go to the doctor. The doctor has to think of that disease. They have to have a test even available that they could order, and then order the right test for the right timing of the person's course of infection. So by the time we get notified about something, and our county health departments are the ones that do the investigations, and I'm up here at the state office, we've already overcome several barriers to even finding out that this person exists. Our county health departments, like I said, are the ones that do the investigations. So they're getting, medical records, they're interviewing the person in order to kind of get those key risk factors. Has the person recently traveled? Do they recall mosquito bites? What were their symptoms? What was the timing of everything? Does everything kind of make sense with the information that we have available? We also regularly have samples that get forwarded to our state public health laboratory, including from commercial laboratories, so that we have samples that we can do confirmatory testing on. And then, of course, nowadays we add on the sequencing for some of them. And then our county health departments are the ones that have that direct relationship with mosquito control. They're notifying of everybody we have under investigation, even if we haven't finished the investigation, even if we don't have like all the lab test results in, even if we haven't confirmed the person as a case, so that they can do their response activities and try to control those mosquito populations. Interestingly enough, though, only about 5% of the cases we investigate in Florida are for arboviruses that are endemic to the United States. So when I'm talking about an endemic arbovirus, think of the things that maybe you hear about most often, like West Nile virus would be kind of the biggest one, St. Louis encephalitis virus, Eastern equine encephalitis virus, someone brought up last week. Those would be the ones that are endemic diseases. We do have those in Florida. We spend very, very little time actually working on those. Over 95% of the time, we are focused on the more exotic arboviral diseases that are being potentially brought in by travelers. So think of things like dengue, Zika, chikungunya, things like that. And so for this presentation, I'm really going to just focus on dengue and Zika just a tiny bit. But we've kind of historically had high numbers of imported cases, a lot of travelers, especially with dengue, coming into the state. And they both can cause this really non-specific febrile illness where the symptoms just sound like a lot of other things, again, kind of making it hard for those healthcare providers to pick out specific diseases. There are a few differences. With dengue, you can have some more severe kind of clinical presentations known as severe dengue. You are at more increased risk of severe dengue if you've had dengue more than once. That's one of the several different risk factors. And then as Nate Gruba mentioned last week, there's also multiple serotypes. So there's dengue 1, dengue 2, dengue 3, dengue 4. And if you got dengue 1, then you're immune to dengue 1, but you could still technically get the other 3. So you can be infected up to 4 times. With Zika, there's also a high rate of people who don't develop any symptoms at all. There's also the risk of sexual transmission. There's been shown to be linkage to birth defects such as microcephaly, and then also some autoimmune conditions such as Guillain-Barré syndrome. So we don't want any of these to be in Florida. And so for the transmission cycle of both dengue and Zika, it's maintained between mosquitoes and people. So it's going back and forth between people, mosquito, just like that. And most cases are actually acquired while a person is traveling to abroad to different areas that have these diseases. And then they can potentially bring it back. And the primary species that are involved with transmission of the mosquito species, Aedes aegypti in particular, loves to breed in containers, likes to bite people during the day, loves to feed on people, and doesn't really fly very far either. And so this is kind of just looking at our travel-related cases over time, just for you to kind of get a sense. I purposely don't have numbers on this slide because I want you to look at the trends. But if you look at the bottom where dengue is in that kind of green color, you can see that dengue is the one that we deal with the most often. And that's largely because there are those multiple different serotypes. And so it really allows for this continuous transmission in endemic areas as kind of the serotypes that are predominantly being transmitted get swapped out. A couple of key things on here. 2019 was a fun year. That was when we saw a change in serotype coming from Cuba, where it switched over to dengue 2. And then 2022 on, basically from there, was a large numbers of travel-related cases associated with dengue 3 in Cuba in particular. And Nate mentioned that in his talk last week, that it was essentially a different lineage that came in and started spreading from Cuba, and then led to large increases in the number of travel-related cases. So the last few years have been a bit busy, to say the least. That last kind of bubble there is over 1,000 cases of travel-related, so if that gives you any sort of indication. It kind of acted like you had a new virus being introduced to an area. So if you look at Zika back in 2016, that was really only 100 more cases than what we saw last year for dengue. So with Zika, you can only get it once. So you have this initial introduction to the Americas at the end of 2015, spread like wildfire. We had a large number of travel-related cases. And then over the few years after that, we largely haven't seen anything since because a lot of people were previously exposed. So it kind of decreased that transmission. And Nate also mentioned last week, Cuba is our #1 source for these travel-related dengue cases in particular. But if we really, more than half come from Cuba alone for us. But if you look at this map, it's really showing the top 5 are all in the Caribbean. So Cuba, Puerto Rico, Dominican Republic, Haiti, Jamaica, those are our biggest ones. So if we have an outbreak in Cuba, we know that we're going to be impacted in some way in Florida, though the Americas in general tend to be where most of our travel-related cases end up coming from. And why is that? So the #1 reason why people are traveling to these countries is because they are going home to visit their friends and relatives. And they're taking the same mosquito bite precautions while they're traveling as they do at home, which is, that they're not. So because we have this right population of travelers that are coming back and forth from these endemic areas, we do have the right species of mosquitoes here in Florida. Dengue used to be endemic until the 1930s, so we've got the right environmental conditions. We can have local transmission. There we go. And hopefully you can appreciate, again, that dengue tends to be our repeat problem. We've had a local case of, one of these diseases almost every single year. We were lucky with a couple of years of not having any local dengue identified. We have had some outbreaks. We had an outbreak from 2009 to 2010, 2013, 2020 during COVID. And then we had an outbreak in 2023, 2024, and we are currently experiencing an outbreak right now in 2025. We'll talk a lot more about those kind of details, but you can see that 2023 was our busiest year. We had just under 200 locally acquired cases, which is kind of similar to what we saw in 2016 volume-wise, where we had 300 locally acquired cases, but then again, we weren't seeing those infected travelers coming back into Florida, so the local cases of Zika were just in 2016 and in 2017. So where are the counties where we see this? So more than half of our locally acquired dengue cases come from Miami-Dade County alone. They have a high Cuban population. They have a high volume of people coming in and out, lots of travelers coming through there, lots of people with ties back to those dengue endemic areas. South Florida kind of tends to be a repeat hotspot for us. As you can see here, overall, we've had 16 counties with local dengue transmission. This map is from 2024, so I have one more to add to the list. But what's interesting is that over the last few years, since 2022, we've actually added 9 counties, primarily ones located in Central Florida. So it does look like the risk patterns in Florida are switching it up a little bit, but Miami still kind of is the one that we see most of the activity at. And specifically for Zika, we also kind of identified these areas where we had this ongoing active transmission. So essentially, we had kind of geospatial clustering of cases spread out in space and time, where we saw about half of the cases in Miami were within these little zones. And because of the risk for pregnant women, these were actually publicized, and CDC issued travel advisories to these specific locations. And so that's really going to come into play for one of our examples later on. But these were areas where we had local Zika transmission, and we had kind of like this continuous transmission, short-lived for a few months in each of the different spots, but they were kind of like a hot spot if you want to think about it that way. Okay, so that's our background. I think that Nate did a very good job last week. So if you didn't watch that presentation, highly recommend it. Talking about how dengue sequencing can tell us a lot of information about what's happening at kind of like this big scale picture. How are the different serotypes, genotypes, major, minor lineages being spread around the world? And when we're doing dengue testing at our state public health lab, we're at least getting the serotype information. So we know it's a 1, a 2, a 3, or a 4, which is useful because if you see a change in that predominant serotype, you know things might get a little busy on the imported case front, or say you had like 2 local cases and they were different serotypes, you automatically know that they have nothing to do with each other, they're unrelated. I think that Nate's lineage tool is going to help provide some additional context to that kind of big picture view. But I think the big question is, how can we more effectively use these trends, the dengue sequencing in general, to inform what we're seeing locally and how we take actions as public health? And, we only started doing sequencing regularly in-house in the middle of about 2023 or so. So we're still learning the best way to apply this information in more real time. A lot of what we have done in these examples I'm going to be going through are retrospective. There's lots of different competing priorities for public health, which all you public health folks on the line are well versed in. But I'm going to walk through some examples of how we've used sequencing in the past, primarily from work we've done with either Nate or with CDC. I know Nate joked last week that public health might say, so what about phylogenetic trees sometimes? But I'm going to show you some phylogenetic trees because I think they help to answer really specific questions that we have. And that's, in my mind, one of the biggest ways that we can better operationalize the use of sequencing to inform what we do, is going in with specific questions that we might be wanting to answer. Whether or not the question is, why are we seeing so many imported cases all of a sudden, like Nate had mentioned with that new dengue lineage sweeping through the Americas, or what can sequencing tell us about our own local dengue transmission? So for example number 1, this was actually back in 2019. We were seeing increased kind of dengue cases coming from Cuba, primarily due to a switch in the predominant serotype to dengue 2. And we unfortunately had a fatal case. This individual had actually traveled with family members to Honduras. They developed symptoms 5 weeks later, which would actually not be consistent with being exposed in Honduras because the incubation period for dengue is 2 weeks. The timeline could be consistent, however, with other family members bringing the virus back from their trip to Honduras if they were bitten by mosquitoes locally, and then that mosquito went on to infect the case. And so, unfortunately, none of the family members had symptoms, which doesn't mean that they couldn't have still been a source of the virus, but they were uninterested in getting tested to prove that they had had that past dengue exposure. And our other question is, well, did maybe the person just for some unknown reason have a really long incubation period that we wouldn't expect? And so CDC assisted us by sequencing some of our other samples from Honduras as well as some from Cuba, since that's where we were seeing most of our imported cases coming from. And you can see from the arrow here that sequencing showed that the fatal case belonged to the Asian American genotype of dengue 2, but it was clustered on the tree with cases coming from Cuba rather than from Honduras. So, what that indicated to us was that the case was likely exposed locally from a strain of the virus that was brought in from Cuba rather than having anything to do directly with Honduras. So one of the things we do now when we have a local case that we're able to sequence is we look to see where it falls in the tree, where could the introduction of the virus have been potentially brought in from. Okay, example number 2. To set the stage, we've had 2 dengue outbreaks occur in the Florida Keys. One was back in 2010 in Key West, which is kind of like the bottom of the Keys, and then one in 2020 in Key Largo, which is like the top of the Keys. Um, Both were dengue 1, so the question came up of whether or not they're related, if they could have originated from the same introduction. Now, decade's a really long time, so it makes sense that they're probably separate, right? Rather than something ongoing, continuous that went undetected. But we did work with CDC to confirm that yes, they were not related. The Key West outbreak was from the Central American lineage, right here. And then the Key Largo outbreak was actually from the Caribbean lineage over here. So not related to each other. Which brings us to example number 3, looking at what happened in 2022 when we started seeing that increased dengue 3 activity coming from Cuba. That year we had over 900 travel-related cases. We had 68 local cases among 4 counties. 59 of those local cases were dengue 3. And so the question was, especially for Miami-Dade County, where it made up the majority of those local cases, was the activity we were seeing the result of multiple introductions that were maybe short-lived and died out without going through these multiple cycles of transmission? Or maybe you had one or a handful of introductions that just kind of took hold and kept spreading, resulting in more sustained transmission. And we did this work with both NATE and CDC, which showed that the local cases in red were kind of interspersed among the travel-related cases shown in purple. So what we like to see that because it indicates there were likely multiple introductions that were short-lived. And one thing to keep in mind, though, is that we did definitely oversample for Cuba, since that was most of what our travel-related cases are. There was one right here, I don't know if you can see it, one grouping of local cases on the phylogenetic tree, which when you then look at where those cases were located spatially, they did belong to a small cluster of local cases identified from the same neighborhood. Now we regularly map case locations, so we already spatially knew that those cases could potentially be linked, but it's nice to have that kind of genetic confirmation to prove, yes, they are related to each other. And just to take a moment to further reiterate that difference between sustained and sporadic transmission, we have lots of travelers that are coming in from dengue endemic areas. Not all of those are going to result in, subsequent local cases. You'd have to be bitten while you're infectious to mosquitoes. That mosquito would have to not only be the right species, but it would have to live long enough to then go through its own incubation period and then go on to bite another person and transmit the virus for you to even have local activity. But when that does occur, the question is, will the cycle keep going where the newly exposed person then gets bitten, mosquito bites someone else, and so on and so on, leading to kind of like this chain of local transmission? Or are you going to have kind of this more sporadic where you might have a traveler lead to one case or 2 cases, and then a different traveler will lead to another case, etc. And they're more of like separate introductions. And one of the biggest questions we get is whether dengue is endemic in Florida, which you would essentially need continuous transmission to maintain that. Or are we non-endemic and just kind of primarily have these short-lived introductions? And that last example I gave would actually support this kind of, non-endemic, several introductions side of things. And we've been working with Nate on a larger analysis of all of like the local cases that we had in our freezers, which really is kind of further demonstrating and proving that we are not endemic and it is new introductions each year that are causing our local cases. And you might remember from last week that Nate mentioned that there was some sustained transmission during 2023 in Miami that we found. I didn't include that trace since he had kind of already gone over that. But one of the biggest things that sequencing really helped to show was that by early 2024, those transmission chains had stopped. And so that's one of the other things we pay attention to when we have a local case. Does it match something we've seen previously, or is it something that appears to be more sporadic? Are we seeing clustering in space, time, all of the above? And for our last example, I'm just going to switch it up to Zika. We would essentially do all the same things if we had a local Zika case. That's what I've been talking about for the dengue cases. This example goes back to our outbreak back in 2016, some retrospective work we did with Nate to evaluate kind of our outbreak afterwards. At the time, we had local cases in 4 counties, and sequencing showed that the Zika was likely introduced from the Caribbean, which makes sense, similar to kind of dengue. It showed, as you might expect, that different counties were likely different introductions from each other, which makes sense because, some of these counties are very far apart spatially. And that essentially there were 4 distinct lineages, and you can tell by them being labeled F1, F2, F3, and F4 in this figure. 2 of those lineages just had a single case. And then if you look at part B here, there's the F1 and the F2 lineage shown in more detail, specifically looking kind of more into Miami-Dade County. But what I want you to pay attention to is the timeline on the bottom here. So in order for the 2 lineages, so F1 and F2 here, to have as much genetic diversity as they have, you would have had to have them been introduced over a year prior if it was just a single introduction. Now that doesn't really make a whole lot of sense epidemiologically. So more likely there were multiple introductions in order to achieve this kind of level of genetic diversity that we're seeing. Now you could say that this was one introduction maybe, and then this was one introduction, but the local cases would have had to have started in March or April for this to have been just 2 separate introductions, which was a couple of months before our first identified case. What's probably even more likely than either of those scenarios is that both of these are probably the result of several introductions rather than 1 or 2 introductions, and not necessarily a large chain of transmission for each of those lineages. And so when we break down the phylogenetic tree, looking at some of those areas of active transmission that I was talking about earlier, we identified those areas by mapping, looking at cases in space and time. You can see that for the Wynwood in blue and Miami Beach in green, the different lineages are actually being passed back and forth between both of those locations. So you're not seeing like a single lineage making up an active area of transmission. It's spread out. And they're just passing it back and forth. So they were not having lineages confined to specific locations, if that makes sense. So spatial clustering can provide you some information, but it might not always tell you the whole story. People are the ones that tend to move these diseases around, less so than the mosquitoes, because the mosquitoes, if you recall, don't fly very far. And in fact, Nate used some modeling to estimate that to have as much genetic diversity again, as we're seeing here, taking into account different possible, reproductive numbers, underreporting, stuff like that, there was likely 17 to 42 separate introductions that actually led to what we're seeing here. That's a lot. So we have kind of like these tools for finding cases. We're doing a lot of interviews, we're getting medical records, but I think genomic epi really helps to serve as an additional tool in our toolbox to kind of help explain, contextualize, aid into some of our investigations. We did also work on a paper with Nate and others that were interested in how genomic epi could be used as a public health tool for mosquito-borne diseases. This wasn't limited to just dengue though, it's mosquito-borne diseases in general, but here were just a few different examples that we came up with. Some might be disease or situation specific, but I think there's quite a few different use cases of value. There's more and more public health labs that are starting to bring in, this in-house sequencing capacity, adding on more mosquito-borne diseases. I know we would like to onboard some more ourselves and improve some of the different things we do right now. I do think there's a lot of room to grow, but I think opportunities like this training have actually been really helpful in bridging some of the gaps between the different disciplines, knocking down those silos and whatnot. So again, not a laboratorian, not a bioinformatician, not even a genomic epi, just an epi that thinks this stuff is cool. So thank you for inviting me here to talk a little bit about what we do. And then here is my contact information.